Hello, good morning, students. My name is Vinit Kumar Singh, Assistant Professor, School of Pharmacy, BBD University, Lucknow. Welcome back, all of you. In previous lecture, the part 1 of anti-malaria drugs, we have studied that the different different species of mosquitoes who involved in spreading this plasmodium, like plasmodium vivax, plasmodium falciparum, and also we see the lifespan and life cycle of the malaria parasite. So today's this lecture we will discuss the remaining part of this topic. So let's start, and hope so you getting some information regarding this. So quinine, we have already studied that cinchona bark, one of the vital plant which play a good role, major role in treatment of malaria in ancient time as well as today also. From cinchona we got a lot of medicines, lots of active constituents which are still active and widely used in different kind of diseases. So structure, activity, relationship of quinine. So as you see that there is a basic structure of a quinine. This quinine structure, okay, so here you can see the structure of a quinine, the basic structure of quinine, and the different groups are added, and where there can be added this different groups is a position R1. So we will study the each part of this ring one by one. The first and major part of this ring is the major part of this ring. Is a two ring composed to each other, and the numbering start from nitrogen compound 1, then 2, 3, 4, 5, 6, 7, 8. Then the position, carbon position number 6 and R1. It's a it's a position where can we change a lot of groups can add and get respectively activity. So quinoline R ring. Presence of methoxy group in quinine is not essential. इसकी यहाँ पे अगर हम methoxy group add कर दें तो that is not essential, and that will not give the good desired pharmacological activity. Replacement of methoxy group by a halogen, especially chloride bromide, these are halogens. क्या होगा ये? That enhance the activity. So enhance the activity of quinine ring, and it's like a further increase in activity resulted from the introduction of phenyl group at position 2 dash. At the position number 2, as you see that position number 2, if talking about the position number 2 right now, then please ensure that I'm talking this ring only, not all. I'm talking this ring only, and this ring only. So this is a N from the starting. This ring, particular this ring, the activity. So further increase in activity resulted from the introduction of phenyl group of at position 2. So पोजीशन नंबर 2 पे अगर हम फिनाइल ग्रुप ऐड करेंगे तो क्या होगा, एक्टिविटी अच्छी आएगी, दस गुड एक्टिविटी. एंड द मिथॉक्सी ग्रुप इट्स नॉट एसेंशियल कि उसमें ऐड करना ही है. that's not that's not a big task to deal with the pharmacological activity. And then ethoxy group would be changed by chloride, just like halogens, especially chloride or chlorine atoms that enhance the activity. And it was discovered that high activity without phototoxicity could be attained by blocking position 2 with a trifluoron ethyl group, a finding that eventually lead to development of efloquine. So the position number 2, it's plays one of the great role. सो यहाँ पे क्या है ना कि द स्क्रीनिन की जो एक कह सकते हैं कि एक ड्रॉबैक है या एडीआर है ये, कि फोटोटॉक्सिटी बहुत कॉमनली ऑब्जर्व की गई है, बहुत कॉमनली इसको देखा गया है कि होता है जो भी पेशेंट इसको लेते हैं. तो इसको टैकल करने के लिए, सप्रेस करने के लिए वाया द SAR टेक्निक, द ग्रुप ट्राइफ्लोरॉन ethyl is added on position number 2. सो उससे क्या हुआ कि इसकी फोटोटॉक्सिसिटी ऑलमोस्ट रिड्यूस हो गई, बहुत कम हो गई. इसकी जो एक एडवर्स इफेक्ट आता था उसकी फोटोटॉक्सिसिटी okay. So it was discovered that high activity without phototoxicity could be attained. So क्या होगा कि यहाँ पे ट्राइफ्लोरोन इथाइल ग्रुप जब ऐड करेंगे तो ये सप्रेस हो जाएगा, और ये इफ्लोक्विन जो डेवलप हुई थी ये इसी का एक बेटर प्रोडक्ट था. देन फिर बात करें हम द पोजीशन नंबर 8 ऑन द अपर रिंग. So activity usually enhanced by the introduction of halogen at this position. So position number 8 क्या होगी, यहाँ पे अगर हाइड्रोजन हम ऐड करते हैं तो एक्टिविटी बेहतर आती है, एक पहले से एक अच्छी एक्टिविटी इसमें शो होगी. Then quinidine portion of the quinidine portion is not necessary for activity. However, an alkyl tertiary amine attached at C9 in portion. The C9 portion is this. Asymmetry at this position is not essential for anti-malarial activity. So doesn't mean that there is a asymmetry or not. That's not a big deal with the antibacterial activity. And here the position number 9, the modification of the secondary alcohol at C9 through oxidation, esterification क्या होगा, that diminishes the activity. So at the position number 9 should not modify or with the oxidation or esterification that lead the diminish of the activity. The configuration at position 8 and 9 affect the just position of the hydroxyl group and the non-aromatic nitrogen atom, a relationship that is associated with anti-malarial activity. So these are the some position like 8, 9 in upper ring, 2 position number 2 dash in lower ring. These are the some very essential part, a very essential position where the replacement or exchange of group can define the new activity. Okay, so this is the basic idea of the SAR of quinine, herring, and the next category of antimalarial drug is biguanides like proguanil, cycloguanil. These are the key drugs, main drugs which categorize under this biguanide category. It is an early example of a prodrug. It is a slow-acting erythrocytic zygenticides which also invade the pre-erythrocytic stages of Plasmodium falciparum. Gametocytes exposed to proguanil are not killed but fail to develop properly in the mosquitoes. It is cyclized in the body to a triazine derivative like cycloguanil, which inhibits Plasmodium DHFR in preference to the mammalian enzyme. Resistance to proguanil develop rapidly due to mutational changes in the Plasmodium DHFRase enzyme. So जो ड्रग है ये क्या होता है, ये एक तरीके से इसको प्रोड्रग के रूप में कन्वर्ट किया जाता है. ये एक बायो ट्रांसफॉर्मेशन होने के बाद ये एक्टिव ड्रग में कन्वर्ट होती है एंड इसका रेजिस्टेंस भी बहुत जल्दी हो जाता है क्योंकि प्लाज्मोडियम डेजोफारेज एंजाइम की वजह से उसको टैकल कर पाते हैं. और लेकिन ये इसकी एक्टिविटी अच्छी है लाइक साइजेंटोसाइड पे भी और प्री एर्थोसाइटिक स्टेज पे भी एक्टिविटी शो करते हैं. अब क्या जो प्रोग्वानिल है ये क्या है, एक तरीके से प्रोड्रग, इसको साइक्लोग्वानिल में कन्वर्ट करते हैं तो साइक्लोग्वानिल एक्टिविटी शो करती है. ये होता है बाय द हेल्प ऑफ लीवर, हेल्प ऑफ देयर एंजाइम साइटोक्रोम 2C9T. यहाँ पे क्या होगा कि प्रोग्वानिल जाती है, उसका क्या ऑक्सीडेशन हो जाएगा, ऑक्सीडेशन हो जाता है बाय द हेल्प ऑफ साइटोक्रोम CYP जिसे कहते हैं साइटोक्रोम 2C19. फिर इसका स्ट्रक्चर मॉडिफाई होके एक वाटर मॉलिक्यूल बाहर निकलता है, फिर लास्ट में इसका टोटोमेरिक इफेक्ट या शिफ्ट होती है. उसके बाद जो है एक दो ढाई स्टेप जो भी है 1 2 3 4, चौथे स्टेप में एक एक्टिव कंपाउंड हमें मिल जाता है साइक्लोग्वानिल जो कि एक्टिव मेटाबोलाइट होता है आपका प्रोग्वानिल का, और ये एक्टिविटी शो करता है. itself proguanil is not showing the activity, it only can effective when that oxidation is or biotransformation happens and the proguanil converts it to cycloguanil, and then cycloguanil is the active compound. Some adverse effect like mild abdominal upper abdominal like upset pain, omitting, occasional stomatitis, not usually, it's sometime, hematuria, rashes one of the common things, and then transient loss of hair will also very rare side effect like quinine. And the main thing is that this drug is not safe during pregnancy, either first trimester, 3 trimester, or any stages of pregnancy. it is not safe. In fact, as well as the baby who are feeding, they are also not safe. So feeding women or pregnant women should not take this quinine. This is the mechanism of action, like para-amoebic acid plus pteridine. ये डाइहाइड्रोटेरोएट सिंथेसिस से क्या बनाता है? डाइहाइड्रोटेरोइक एसिड, देन डाइहाइड्रोफोलेट सिंथेसिस, इस पे हाइड्रोटेरोइक एसिड से आके फिर क्या बनाता है? इससे डाइहाइड्रोफोलिक एसिड. Dihydrofolate reduces tetrahydrofolic acid and other precursors like thymidine, purine, then ये DNA में बनता है. अब जो ड्रग्स हैं लाइक सल्फोडॉक्सिन, सल्फोमिथोजाइन, डैप्सन, these are the act on the primary enzymes like dihydropteroate synthesis and then dihydrofolate reductase. This enzyme is inhibited by pyrimethamine, proguanyl and trimethoprim. Then the proguanyl, it's then convert transformation. The active compound is cycloguanyl that act on the dihydrofolate reductase and block it. That's why the dihydrofolic acid that not converted in tetrahydrofluoric acid, and further the thymidine, purines and other essential nuclear part will not be synthesized in parasite, and that's why the parasite will die. So this is the structure, mechanism of action of cycloguanyls. Basically this cycloguanyls act on dihydrofolate reductase, the third enzyme involved in this. The next compound is diamino-pyrimidine. These are also categorized. This is a class of drugs. In this class, two drugs are categorized like trimethoprim and pyrimethamine. These have almost the similar structures, two ring in both and pyridine ring in both. And it's slow acting erythrocytic side to side. Then they have a limited or slow activity on side to side. Direct inhibitor of plasmodium dihydrofolate reductase, it's also called a DHFR, is direct inhibit करती है. Then conversion of dihydrofluoric acid to tetrafolic acid is inhibited. High dose inhibit like toxoplasm, and resistance developed by the mutation. The registration has happened by the same means as we see earlier. The mutation of DHFRase enzyme happens, and that does not allow to entry in diaminopyridines derivatives, and they do not bind to their active site, and the remaining part of the synthesis of DNA by parasite can easily happen. So diaminopyridine more potent than proguanyl and effective against erythrocytic forms of all species. Means these either falciparum ho या vivox ho, they act at all kind of species. Then pyrimethamine, adverse effect like occasional nausea and rashes, folate deficiency. The folate deficiency is rare, not very common, and it happens in some amount of folic acid will give to the patient. Then megaloblastic anemia and granulocytopenia with higher dose. If we use higher dose rather than prescribed, then could be it's happen, can be treated with folic acid to, to overcome this ADR. Then combined with sulfonamide or dapsone for treatment of falciparum malaria. Because sulfonamide and dapsone have the same and a similar mechanism of action, like they also act on the same chain process. In fact, the same reaction, dapsone and proguanyls. Then sulfadoxine, pyrimethamine. Subsequential blocked sulfadoxine 500 and pyrimethamine 25 mg tablets once for acute attack. Not recommended for prophylaxis. Effective blood sinusoid against Plasmodium falciparum. Treatment and prophylaxis of falciparum malaria are restricted to chloroquine. So as see, the mechanism, like para-aminobenzoic acid, folate, tetrahydrofolate, synthesis of thymidine etc. Dihydropteroate synthesis, sulfonamides, folate, dihydrofolate reductase, pyrimethamine, then tetrahydrofolate, synthesis of thymidase etc. So pyrimethamine is block this dihydrofolate reductase, and the dihydrofolate and then dihydropteroate synthesis is also inhibited by the sulfonamide. So the point is blocked by drug, and the all kind of possibility will suppress to survive a parasite. Adverse effect like mild GIT upset, megaloblastic anemia, bone marrow depletion. These are the anresis urticaria, serum sickness, drug fever, and exfoliative dermatitis, Stevens-Jones syndrome. Stevens-Jones syndrome, it's quite difficult or quite not easy with patient, but rarely it's seen that after the taking of drugs, some patients have developed the symptoms or disease of Stevens-Jones syndrome. Nephrotoxicity also is very common. Single dose treatment of uncomplicated chloroquine resistance falciparum malariae, like patient intolerant to chloroquine, first choice treatment for toxoplasmosis. And then the next class is 8-aminoquinoline. Previously we studied 4 aminoquinolines. This is the 8 quinines. So primaquine, palmaquine, pentaquine, and the last drug is quinoside. These are the drugs which categorize under this category 8 aminoquinolines. The primaquine, and this is a palmaquine and pentaquine. The basic structure remains same in all the drugs, but the substitution will change accordingly. The most of the substitution will happen on this position NH, NH, and NH. Like primaquine, it is the only 8 aminoquinoline in clinical use. The rest of the drugs like palmaquine or pentaquine, quinoside, these are not used in clinical. These are not clinical use still, and the only drug which are in market or which are used in a clinical purpose, like a primaquine. It is largely used to prevent relapse or falciparum ovals and falciparum vivax malaria by eliminating dormant hypnozoites, and it also has activity against the pre-erythrocytic stages and gametocytes of falciparum. It is not used for prophylaxis. It is its spectrum of activity one of the narrowest of the current use anti-malarial drugs, being indicated only for exoerythrocytic Plasmodium vivax. Not clear. It's the mechanism of action. Primaquine. It's not so clear. It's converted and produce active oxygen interference with plasmodial mitochondrial function. That's not exact mechanism of action is known, but the okay. Okay, sorry, yeah. So exact mechanism is not known, but the most possible, most appropriable the mechanism of action is considered like they interfere with mitochondrial function. Then use the have a radical use like Plasmodium vivax, so will give in acute attack or throughout the incubation period, prevents relapse. Then prophylactically before and after leaving the endemic area to eradicate hepatic form, effective vector control is possible or used in area of low transmission. Falciparum malariae 50 allowed, no, less than 50, around 45 mg with chloroquine used like gametocidal and cut down transmission where effective control is needed. Then adverse effect like therapeutic doses पे क्या होगा कि hemolysis and megalovastic commonly seen in 6-GPD deficiencies, nausea, headache, and epigastric pain, abdominal cramp. It's also the very rare this leukocytosis and agranulocytosis. Leukopenia is a very rare but seen. Then precautions: primaquine should not be given during pregnancy because fetus is G6PD deficiency. जो fetus होता है उसको अंदर 6 glucose 6 phosphate diarase phosphate dehydrogenase 6 G6PD deficiency होती है और फिर वो एक्टिव नहीं रहता. Sulfonamides and sulfones, like sulfonamide and sulfones, there's a dapsone, sulfodoxine, amine alcohol, etamine alcohols, leofenin. These are the drugs. Structurally, halofentrine differ from all other antimalarial drugs. It is a good example of a drug design that incorporates bioasteric principle as evidenced by the trifluoromethyl moiety. Halofentrine is side and has no effect on sporozoites, gametocytes, or hepatic stages. It is active against strain resistance to chloroquine and pyrimethaquine. Cross resistance in falciparum infection occurred. And then erratic viability, lethal cardiotoxicity, and cross resistance to mefloquine limited its use nowadays, used only when no other alternative available. Then the specific adverse effect like abdominal pain, headache. These are the some specific adverse effect. Increase in hepatic enzyme, also cough, and may cause hemolytic anemia, conversion, reserved for infection case by resistance organism. And contraindication with mefloquine, like patient with cardiac conduction interfering in pregnancy. So cardiac ho ya pregnant women, both should not be used, or without consent or without permission of a physician should not be used. The next category of the anti-malarial drug is ascoterapine in lactones. Here is the structure, the photograph of artemisinin tree. The artemisinin series are the newest of the anti-malarial drug and are structurally unique when compared with the compound previously and currently used. The parent compound artemisinin is a natural product extracted from the dry leaves of Artemisia annua. All of the compound given in finger are active against the Plasmodium genera that cause malaria. The key structure characteristics appears to trioxane consist of the indo-peroxide and dioxane oxygens. Not stereochemistry at position 12 is not a critical artemisinin structure of artemisinin. There is the artemisinin derivative used in malaria like artesunate, artemether, arteether, arterolant. Mechanism. This compound contains indo-peroxide bridge. Indo-peroxide bridge interact with heme in parasite. Heme iron clears the indo-peroxide bridge. There is a generation of highly effective free radical which damage parasite membrane by the covalently binding in membrane protein. So artemisinin activation, alkylation, and they act rapidly, killing blood stages of the all Plasmodium species and reducing the parasite biomass. Artemisinins have the fastest parasite clearance time of any antimalarial, and artemisinins are active against gametocytes. The parasites from that is infection to mosquito and their use has been associated with reduced malaria transmission. Safe and 10 to 100 times potent compared to other antimicrobial. Artemisinins के जितने भी होते हैं कंपाउंड या डेरिवेटिव वो प्रीवियसली यूज जो भी हो रहे हैं क्लोरोक्विन जो भी उससे ऑलमोस्ट 10 to 100 टाइम्स पोटेंट होते हैं. एंटीमलेरियल एक्शंस artemisins then the antimalarial actions artemisins artemisins is a very good drug. It's act on both in hematolytic and gametocyte position both. Artemisinin based combination therapy in general artemisins should not be used as a single agent to prevent emergence of drug resistance due to avoid the need of prolonged therapy. Artemisinin based combination therapy combine the highly effective short acting artemisins with a longer acting partner to protect against artemisinin resistance and not to facilitate during conveyance. Acts are typically administered for 3 days and are often available in fixed dose tablet. 4 acts are recommended by the who are the treatment of uncomplicated malaria like artemether, lumetrine, artesunate. These are the very active combination used. Artemisins like intravenous artesunate is used for treatment of several malaria. It is superior to quinine for treatment of several malaria with respect to clearing like parasitemia and reducing mortality. Given the short half-life of artemisins, intravenous therapy must be followed by the longer acting agent once the patient is able to tolerate oral medications. If used along artesunate must be administered for 5 to 7 days. Treatment for less than 5 days result in recurrent parasitemia several weeks after after therapy due to the very short duration of action rather than to artemisinin resistant. So these are the some artemisinins are generally well tolerated होते हैं यहाँ पे. Type 1 hypersensitivity to artemisinin compound has been reported. Some advantage like rapid clinically एक्टिव होती है, हाई क्योर रेट इसका रहता है कि absence of parasite resistance, good tolerability profile and dosing schedule is simple. The naphthalidine. The naphthalidine is a newer drug from micacrine. This structure is mipacrine developed in China. Mechanism similar to chloroquine, high effective erythrocytic cytoside, effective against chloroquine sensitive and resistant vivax and falciparum malaria. Slow onset and long duration of action concentrated in RBCs. Water soluble, dirty half, about 7 days, and oral parenterally used, well tolerated at high dose used analgesic and antipyretics. So these are the some drugs which are widely used in anti-malaria and for the cure of anti-malaria. And I hope this video will benefited and given some knowledge regarding anti-malarial drugs. So thank you very much for listening and giving this time to video. Thank you very much.